Uh, I'll just begin the class. My net is a little unstable today, so I'm keeping the video off for now. Um, okay, so basically we'll do a quick recap of our previous session, and then we'll move on to the new discussion for today. So, good evening. Welcome to today's class. In the last class, we had continued our discussion on methods of studying DNA protein interactions. In the first class on this topic, we had discussed in vitro methods, and then in the previous class, we moved on to in vivo methods. So, when we talk about in vivo DNA protein interactions, what do we mean? We basically want, like specifically, what do we mean by in vivo and in vitro? Just to put it like in a brief manner, in vitro DNA protein interactions are those which you expect to occur if um, both the components are added separately, and the interaction can be something which you can simulate by simple addition of a DNA molecule, a short DNA molecule, and some protein X. However, when we talk about in vivo, it means that within a living system, specifically within a living cell, at any given point of time or under certain conditions or certain circumstances, for example, in the presence of a drug or the absence of it, or when a cell senses some sort of stimulus in response to that, when there is a DNA protein interaction. So basically, it's not a sort of forced interaction or not an artificial sort of interaction. It's something which is occurring inside the cell, which means it carries some relevance. And therefore, depending on what sort of approach you are taking forward in your study, for example, if your objective is simply to characterize whether or not a certain protein molecule can bind to a certain DNA sequence. So you might ask, when is that relevant? Sometimes people design certain drugs which are meant to target certain regions of the genome, or when you want to design however, of course, that won't be very specific, but more importantly, many a times people wish to design certain oligonucleotide sequences which can instead inhibit a protein which is causing some harm inside the cell. So basically, you are competitively inhibiting the ability of the protein to do its job. So when you do that, under those circumstances, you want to under in vitro conditions, you want to validate that interaction. However, in vivo is more relevant when your idea or when your objective is to study a biological interaction between a DNA protein, between a DNA molecule and a protein sequence. Now, when we talked about in vivo techniques, there are several methods, and the DNA inside the cell also exists in a specific conformation, which we have discussed previously, whereby it assumes um, a nucleosome threads on a string sort of architecture. And the compaction of that chromatin is especially relevant to ensure that only certain regions of the chromatin are opened up so that control over gene expression can be maintained by the cell. So, yeah. And also, of course, in vitro techniques do not faithfully replicate these conditions. And secondly, in vitro DNA is also naked in structure. It does not consider nucleosomes and the histones, which are forming the nucleosomes, into consideration during the study. Now, one method which we discussed in the previous class is chromatin immunoprecipitation, which is extensively used for epigenome-based studies to understand diseases, to understand simply DNA protein interaction, to identify regulatory axes. For example, if you want to study whether a protein X has the capacity to regulate the expression of protein Y, then under those circumstances, CHIP is a wonderful method to validate the same. And DNA protein interactions are, of course, biologically very relevant in terms of every process, replication, transcription, DNA repair, etc. All of this is executed by proteins, and DNA forms a major component in all of these procedures. Now, the name CHIP or chromatin immunoprecipitation suggests simply that you use the chromatin from a living cell and you use immunocomponents, which are antibodies, to selectively immunoprecipitate your protein of interest and then find out what region of the genome is bound to it. And this is just a representative example of how DNA protein interactions can possibly occur. This is, of course, your DNA molecule, and you see that not all regions of a protein are usually DNA binding in nature. Instead, when you study the secondary and tertiary structure of proteins, you will find that there are certain regions, otherwise called as domains, of which some regions are specifically considered to be DNA binding domains. And we already in the previous class have discussed the different types of proteins that bear DNA binding domains, like there are zinc fingers, there are leucine zippers, etc. So there are different protein families as well. So then CHIP has been used to study largely transcription factors, histone proteins, and more importantly, even CHIP can be used even to study modifications of a certain type present on certain proteins. Like most commonly, this is observed for um, serine to phosphorylated RNA polymerase 2 or histones which bear a specific modification mark. And we will discuss about that today, and various sort of other modifications as well. But these are most commonly the strategies which are utilized. Yeah. So these are most commonly the strategies which are utilized. Now, the technique was initially discovered by David Gilmer and John T.L., who are considered to be the pioneers of this technique. And their study was on the RNA polymerase in bacterial systems, whereby they covalently cross-linked the DNA and protein using UV irradiation. This, of course, is not something you usually will perceive for now. Instead, chemical cross-linkers are utilized. We'll discuss that as well in a couple of slides. And it has already been covered in the last class, which is why I'm uh, rushing by this topic a little bit. But because it's related to the technique we'll be discussing today, so I want to recapitulate what we already did. So yeah, so basically once the DNA protein interaction has been cross-linked using UV radiation, cells are lysed and the bacterial RNA polymerase is immunoprecipitated, and then the bound DNA can be assayed using probes corresponding to different regions of known genes. Later, this was also used for eukaryotic DNA RNA polymerase and not DNA, sorry, RNA polymerase, and Drosophila heat shock genes as well, and they use formaldehyde, which is what I just mentioned. Chemical cross-linker is usually preferred. Now, vaguely speaking, the steps that are involved in CHIP or chromatin immunoprecipitation, A, involves stabilization of the protein and DNA interaction. By stabilization, we basically mean freezing those interactions in vivo. So you do not want it to get disrupted because of any mechanical pressure, because of any of the chemical reagents you are adding, or by sonication, which is a necessary step during chromatin immunoprecipitation. Then followed by cell lysis, extracting the nuclear material, fragmenting it, pre-clearing the solution to remove any non-specific precipitation that might have occurred. And then finally, you elute the DNA protein complexes and digest the protein to finally get the DNA molecule. Now, whether or not you will digest the protein depends upon what your objective is. Most commonly, you would know one of the two components in more detail. So most commonly, you would find for chip sequence, for chip, the method by itself, the idea that is usually opted for is to see whether or not your protein is bound to a region of your interest. And so you want to retain the DNA material and get rid of the bound protein. Again, depending on the context, different optimization might be necessary. So this is what the workflow shows as well. After cross-linking, cell lysis gives you this sort of extract, which you subject to sonication and enzyme digestion. Then you fragment the chromatin and immunoprecipitate with protein with an antibody against your protein of interest. And then what you get as your precipitate is your chip material. Then from there, you purify your DNA sequence of interest and analyze it using multiple methods. Most commonly, you would find qPCR, chip qPCR to be performed as a validation of chip sequencing data. So next we move on to the reagents, which are necessary in this entire protocol. The first step, which we already discussed, whereby we freeze interactions present in vivo between DNA molecules and protein molecules. And for that, cross-linking is the necessary step. So covalent stabilization of this interaction is carried out by a number of different molecules. Most commonly, you will find formaldehyde, like I already mentioned. But there are many other cross-linkers as well, like DSG, EGS, DMA, and NDSS. Most commonly, I think you would find NDSS being used in labs. Um, but yeah, depending on the optimization of your technique, you can choose any of them. Now, formaldehyde, while most commonly used, has certain limitations. It does not maintain all types of DNA protein interactions. For example, if there are co-activator interactions and transcription factors in hyperdynamic equilibrium. Also, it is a zero-length cross-linker, so it cannot accommodate very long-range DNA protein interactions. What do we mean by long range? We basically mean that if it involves interactions between a protein and a DNA, which is physically at a significant distance, then the ability of formaldehyde to work gets limited, and what you might get in that case is a sort of false result, which is of course not very desirable. Now to trap Now, to trap larger protein complexes, which are forming a chromatin loop and forming a quaternary structure, longer cross-linkers are often utilized, most commonly DSG, like I mentioned previously, or EGS. These might be utilized. And you can see next to it, the Armstrong length has been mentioned, um, which sort of indicates the distance which can be accommodated by these individual reagents. And also sometimes two-step cross-linking may also be performed for more robust, more sort of efficient cross-linking. Now, the condition and the timing for cross-linking, that needs to be optimized. But under more standard protocols, you would find formaldehyde cross-linking being done for somewhere around 10 to 15 minutes. And this can also, additionally, also, if you leave it with formaldehyde for an extended period of time, sometimes it can affect the sonication process as well, because beyond a threshold, to some extent, non-specific or random cross-linking may also occur, and that is again going to possibly give you some undesirable or rather false results, which you will not be able to troubleshoot and might contradict your initial hypothesis or might falsely validate your hypothesis. So either way, these things should be optimized over time. And yeah, another issue with cross-linking being performed for an extended period is that it can sequester epitopes and then compromise the ability of the antibody to eventually bind when you perform immunoprecipitation. So you do not want your epitopes to get masked. You want it to be available or accessible to your antibody. So this is just how it looks. You have polymer 1, you have polymer 2, which are getting linked by a cross-linker. This is covalent bonding during a sort of chemical cross-linking. Now this cross-linker is a little bit different. Only a part of it is retained and there can be covalent bonding. Now if you see the difference between these two, you can see this is short range and this is much, much more long range. So depending on whatever literature search you have done, you should adjust your protocol accordingly. Then there can be physical cross-linking also, whereby you do not have any sort of chemical influence, and the nature of that interaction is not very covalent in nature, and it is less stable and more easy, obviously, to disrupt. Then you have enzymatic cross-linking, which is also a covalent form of interaction or covalent form of bonding between these two entities. And that is, again, another possibility that does exist. Second step is cell lysis. So after cross-linking, you lyse the cell. You want your DNA protein complexes to sort of come out into solution. This is achieved obviously by a detergent, which you need to sort of form pores on the cell and then allow the cell lysis to proceed smoothly. And some salt-based buffers are also utilized. Generally, treatment with the chip lysis buffer is recommended, and removal of the cytosolic part is also done during this step because DNA protein complexes are present in nuclear fractions, and usually mechanical lysis is not preferred during this step. Um, Shearing of DNA, when we talk about that, so extracted nuclear part, what do we mean by shearing of DNA? Basically means that the entire chromatin, if you consider the entire chromatin present within the cell, it is immense, it is large, and it is impossible to properly pull down any region of the DNA in that case, because if the genome is that large, then it might either simply settle by virtue of size, or secondly, it might not settle and no pull down can also occur. So instead, what is done is that shearing of the DNA is carried out, or sonication of the DNA is carried out, as a result of which the entire chromatin gets fragmented into little regions depending on what parameters you set. Somewhere around 700 to 800 base pairs is mostly considered ideal. Again, if your protein is very large, you have to adjust accordingly and vice versa. But DNA shearing can either be achieved by sonication or enzymatic digestion performed using MNAs. That's another possibility as well. And DNA shearing is very crucial. And sonication, while it needs optimization and cold temperature, it is quite fuss-free as well. So multiple freeze-thaw cycles should be avoided with the sonicated chromatin. It should preferably be immediately utilized for immunoprecipitation, and then you can visualize the uh, fragmented DNA. So yeah, I think this is pretty much the same thing. Just one thing there, this instrument that I have shown here, which is called as Covaris, is sort of an ultrasonic heater, which uses sound as the form of energy, which is able to disrupt non-specific DNA protein complexes and sort of optimize the output of this experiment. So these are the two strategies. The first one is called as Bioruptor, which is most commonly produced by this company Diagenote. This also uses ultrasonic. This is also an ultrasonicator, which is very commonly utilized for chromatin immunoprecipitation. And the other instrument that I just talked about is called as Covaris. So it uses focused acoustics to shear DNA, and here you can adjust the intensity, the cycles, the duty factor of your instrument, and the time up to which shearing has to be done. Again, how do you know what is the correct parameter that needs to be set? You will have a lot of protocols available online, so you can standardize that based on, again, your requirement. Then this is what the gel can possibly look like after you have performed shearing. So you would expect that if shearing has occurred properly, you would expect that there should be sort of a smear-like structure with no regions that are showing any crisp bands. If that is present, it indicates incomplete shearing has occurred. And also, if it does not migrate through the or move through the gel, in that case as well. Yeah. Next, you can see this is what the output from a covariance instrument can look like. You will see what is the distribution of each of these types of or lengths of curves from 150 base pair to 5000 base pair, and different sort of lengths or stretches of these fragments post sonication. The difference can be noted here. Next comes the immunoprecipitation step, which is again very crucial. Here, chip validated antibodies should preferably be used. Why so? Because chip, if you think about it, is a very resource intensive protocol. And also it is, yeah, and also it is chip qPCR is usually performed after chip-seq analysis once that has been done, and it's sort of used as a validation more common. So these chip-validated antibodies are used for the selection of their target protein, their interactions, and removal of all other sort of protein components. And then you have your antibodies, which preferably should be very specific to your protein of interest, or they can be tagged. Tag most commonly you would find are some sort of flag tags, or let's consider flag tags only. Then in that case, you can utilize flag tagged beads, and from there you can elute your region of interest. So yeah, these are different possibilities that can permit. And most commonly, you would find chip qPCR being performed to validate the results that you have that you expect. Yeah. So this shows you pretty much that. Only if there is any doubt, please stop and let me know because I cannot hear you at the moment. But just let me know and I'll try to explain it to you better again. Next comes the reversal of crosslinks and DNA characterization. Uh, Crosslink reversal can be done through heat incubation, proteinase K treatment, and protein DNA interactions. If they are tough, then high salt treatment may also be used before proteinase K. And then the DNA may be purified by phenol chloroform using DNA purification kits or columns. And finally, you analyze the DNA sequence. Just a minute. Just give me a minute. Yeah, sorry for the disturbance. Finally, you analyze the DNA sequence that is present using PCR, chip qPCR, or amplification by chip on chips, sequencing, or cloning techniques, etc. So yeah, this shows you again the same thing. Then I showed some variations, X chip, which is most commonly used. I'll not get into that. You can read it once I share the slides. Then we have native chip, where proteins are which are present in their nascent condition, uh, bound to the chromatin. Like most commonly example that you would find are histones that have very high DNA affinity, and hence these do not require any formaldehyde cross-linking. And the native chromatin within a cell ends up producing very small fragments, which by MNase treatment can be immunoprecipitated using an antibody specifically against the protein of interest. This technique is quite mild. It does not result in loss of antibody epitope during immunoprecipitation. So yeah, this is another method. Then you have carrier chip. Most commonly, carrier chip needs to be performed when you have very few cells and say somewhere around 100 cells, and therefore it's useful in such circumstances. Most commonly, you would find that if you have some patient tissue, like basically something that cannot be replenished. So immunoprecipitation of such a yeah of such a small amount of chromatin is facilitated by the addition of carrier chromatin from Drosophila or any other species, which is evolutionarily very distinct from the species that you are investigating to provide effective chromatin precipitation. Here, native chromatin becomes partially digested and immunoprecipitated, and the low amount of chromatin is detected by radio PCR, phosphorimaging, etc. Anyway, you can read about it. Basically, the idea is that when you have very little amount of chromatin, then many a times an extra or additional amount of carrier DNA can also be added. So that's what we saw. Then I think this was our last slide for the last class. Finally, we talked about chip, which sometimes also is known as chip on chip. So it combines chromatin immunoprecipitation with microarray, and you label basically your pull down DNA fragments with a fluorescent dye and then combine it with genomic DNA label with Side3 as reference. Yeah. This probe mixture can then be applied to the microarray chip. Usually it consists of whole genome and then you allow hybridization. The results signify the regions of DNA which become enriched, and this is obtained most commonly in the form of sort of a one-dimensional series of signals with peaks identifying those regions which are bound by protein of interest. Additionally, since the exact location of each element is known, a genome-wide map of DNA-protein interactions can be constructed. It has advantages like in chip-on-chip, you can actually probe a large number of genomic loci at the same time in the same experiment. which helps you to sort of eliminate bias and save time. And secondly, you can also use publicly available data sets to study this. And thirdly, you can analyze multiple genes at the same time in many different classes and then carry out their statistical comparison. So yeah, I think that's where we ended. Yeah. So just let me know if there are any doubts, and then we'll begin the session in a minute.
Ma'am, I have read about it, so like it was clear to me. Like
Oh,
the X chip and those portion were new to me, so I will just go through it once. But it was quite familiar.
okay, okay. So next we'll move ahead and we'll understand how now the what we discussed till now is something you do in the lab where you have cells and you do the pull down and you just end it at that and then you analyze it using qPCR. However, when you want to like the idea behind a qPCR or basically targeted studies is that you have a hypothesis in your mind and you want to specifically look at one or a few regions which you expect to be regulated by the protein that you have pulled down. However, when you talk about chip sequencing, the idea is completely different. The idea is to study the protein that you are pulling down and see every possible regulatory role of that protein within a cell. So the view is a lot more global in nature. The data has a lot more possible implications that you can or applications that you can use it for. And thirdly, it gives you a repository that not just you but anybody, once the data is uploaded, anybody can utilize, and more information can be known about cells. And additionally also it is quite relevant if you expect that your protein of interest gets disrupted by some sort of a treatment. And say, for example, if your protein of interest is a chromatin remodeler and you are using your drug of interest and that is sort of knocking down that protein or just eliminating that within the cell, transcription should completely get disrupted. Chromatin loop should be, lost, and a lot of other downstream implications are expected. And once you have done that, you can also combine it with other methods, which is something we'll do around our last few classes, where we'll understand how integration of these multiple high throughput methods gives us information that is biologically relevant. So when you combine it with RNA-seq data and you perform pathway analysis, you will get more relevant information, which you can eventually connect to formulate your entire story. So coming to chip sequencing, so chromatin immunoprecipitation, which we already discussed, followed by sequencing and analysis is a key technology used in epigenomic research. Again, this method also utilizes an antibody which will bind to your protein of interest, and you expect it to be a DNA binding protein with a preferred locus or a preferred site. And many a times also the other possibility exists that you are targeting not a protein of interest, but rather a modification of interest. And among them, histone modifications are most commonly used, and they are used to identify different locations which are enriched within the genome. And also, why is this relevant? We'll discuss about it more in the next couple of slides. But if you remember our initial discussions on epigenetics and nucleosome formation and histone modifications particularly, then we had discussed how different modifications have how they are read as different signals inside the cell. Some of them are activating, some of them are repressive. And even among these activating and repressive marks, there are subtypes, and all of that information can be understood very well using ChIP-seq. Uh, yeah, so eventually the large amount of data, and data in this case is quite large when we talk about ChIP-seq. But this large amount of data plays an essential role in understanding transcriptional regulation and histone modifications. They dissect the characteristics and the biological functions of certain epigenetic signatures which might be of your interest. And NGS has made this entire process a lot more smooth and a lot more, uh, well, let's call it affordable in today's time, and making it a doable technique in the recent past. So, yeah, if you look at the figure here, you would see that chromatin, which is arranged in this sort of beads on a string structure that we talked about, when that is bound to proteins, in this case, the example taken is P53, protein of interest, the master regulator of the genome, then when you use an anti-TP53 antibody, it selects only those regions which are bound by the protein, and as a result, you will get the DNA which is bound to it. And then you purify your DNA fragments, you remove any unbound chromatin and proteins, as you see here. Yeah, and then you use the purified DNA fragments for sequencing, and then eventually you map it to the genome. This is the like the most relevant, the most biologically informative part, because just getting a random fragment is not good enough, but understanding where that fragment has come from, which kind of is like alignment, which we have already discussed, when you align your read to the genome. And then you expect that because of the difference in the number of fragments which are able to bind to that particular region, the intensity or the efficiency of your protein X to bind to that region may vary between condition 1 and condition 2. Don't worry, we'll learn more about this in the next few slides. So these are the 5 core histone marks which were proposed by Roadmap Epigenomics Consortium and is extensively used for chip-seq analysis. What are these? First, you have H3K4ME1 and H3K27AC, which is associated with enhancer regions. So this is sort of an activation mark. Next, you have H3K4ME3, which is associated with promoter activation. Then you have H3K36ME3, which is associated with identifying which regions have active transcription inside the gene body or within the gene body. Then you have H3K27ME3 or trimethylation, which is associated with polycom repression. H3K9 associated with heterochromatinization. H3K9ME3. So basically, you do not have to sort of memorize or mug up all of these things. But basically, the idea is to convey that A, depending on the histone, B, depending on which residue on the histone, and C, what sort of modification is present on that residue of that histone at that particular point of time. And this can vary also. Uh, yeah, so pretty much that is what we expect. And majorly, this is more relevant when you are comparing a wild type and a treated condition. You must make sure that both your samples are treated for the same duration, and that makes the comparison more relevant. So this is what you see here on the left. You see a distribution of histone modifications, which are across transcriptional regulatory elements of active and silent genes in T cells. Characterizing chromatin signatures in immune cells have been performed using T cells, and these have been derived from the human T cell ChIP-seq data. So enhancers typically are characterized by the presence of H3K9, H3 acetylation, H3K4ME1, and H3K9ME1 in T cells. And then CpG islands are localized in an approximately 1 kb region around the TSS, and they tend to have high acetylation. This is spread out evenly with very low levels of H3K27ME3. Basically, a lot of things have been said in the past 2-3 minutes. But what do I want you to take away from here? I want you to remember that there are 5 core histone marks, not just the histone mark, but the modification present on a particular residue um, is something that can determine what sort of effect it will have. And therefore, individual sequencing experiments can be done. Just a second. Yeah. So this is your this is a comparison between an active gene and a silent gene. So how does the ChIP-seq data reflect this fundamental difference? So if you see here, you will see there is a difference in the mark which is present, and the intensity of the mark in the active gene is much higher versus in a silent gene. You would find these regions to not get to not be activated at all. So depending on your requirement, this might happen. Yeah. Next, you have ChIP-seq overall workflow. Once again, the first step begins with proteins that are binding to genomic DNA. Yeah. So second step is cross-linking. Most commonly, like I mentioned previously, it is a chemical cross-linker like formaldehyde, DSS, DSG, etc. And following that, you have DNA shearing today. Not today. DNA shearing that needs to be performed. And shearing, while sometimes is called as fragmentation, preferably use the term shearing. Then, after your DNA has been sheared, you have to pull down your protein of interest. You expect the bound DNA to be pulled down, following which you will perform DNA purification, sequencing library preparation. Most commonly, this is usually sort of shipped or outsourced. But if you have the facility for it, you can proceed with the same as well. And this involves ligating adapter sequences at both ends so that your sequencing library may be constructed. Yeah, next you have NGS sequencing, you map the assembly, and then you trace back where exactly these XYZ fragments have come from. And then you identify the motif to which this particular protein binds, and you will find multiple databases which actually do this job, where they carry out motif analysis or motif studies to identify whether there is a pattern between this. So sequence analysis and motif identification is going to help you identify peaks, indicating that there is um, binding of protein, binding of a particular protein in a certain region. Just give me a minute, I will be back. Okay. Okay. So yeah. So after cross-linking and fragmentation, okay, I didn't mention, I mentioned MNAs in the last class, but yeah, basically you can either proceed with sonication or micrococcal nucleus digestion. But yeah, depending on your protocol, you have to choose the same. Following which you will perform the actual chromatin immunoprecipitation for your protein of interest. If chip PCR or ChIP-seq or cut and run or IP are part of your eventual objectives, before purchasing your antibody, you should always make sure that the antibody is of this grade. When we say this grade, we basically mean that the epitope which this particular antibody is able to bind is something which is exposed and is not just present in a completely denatured protein, which is usually expected when you perform Western blotting. So a Western grade antibody will probably may or may not work for chromatin immunoprecipitation, because there you completely get the protein sort of turned inside out because of the denaturation. However, for chip, that's not going to happen. You want the protein to remain in its native state. Next, you have reversal of the cross-linking step and purifying the DNA. Your protein, RNA, all of that is digested. DNA gets purified, which represents the sequences which are bound by the protein, and then you can send it for sequencing. Now, for starting the sequencing protocol, like we saw in that image, you have to construct the library. So, end repair can be performed on the purified DNA, and then you attach your sequencing adapters, and then these are slice selected using gel electrophoresis, and then you can create your sequencing library. And finally, sequencing. If you remember, we had already discussed this, I think, when we began our classes on high throughput sequencing method. We talked about different strategies which are often used for sequencing. Now, in this case, it is simply DNA sequencing, and it can be performed using any available NGS platform like Illumina, Solid, Helicos, etc. And this involves different methods of amplification, sequencing by synthesis, allowing the massive parallel sequencing of DNA fragments. So, yeah, this is additionally something else that I wanted to show the comparison between ChIP-seq and chip, which we already discussed. So obviously, first of all, the method is different. You use DNA hybridization on probes which are present on microarray chips, hence the name chip. And ChIP-seq involves immunoprecipitation, which is followed by high-throughput sequencing using next generation methods, which is going to give you much more comprehensive and much more extensive results. Second, the genome coverage, which is limited to sequences on the array. So only some specific genomic regions might get analyzed. Secondly, it allows for very comprehensive coverage, including repetitive regions that might get missed by chip that is taken care of by chip-seq. Especially repetitive regions usually are not considered on the microarray tile or the tile microarray. Then you have cost effectiveness. Chip on chip is more cost effective, but chip-seq, obviously, because it's more high throughput, is more expensive. Resolution, very limited. It depends upon how broad spectrum is the range of the probes which are present on your microarray chip. However, for sequencing, you will have as much as single nucleotide resolution as well, which will give you very precise mapping of binding sites on the genome. And finally, for ChIP-seq, the amount of DNA required is very low, but for chip-on-chip, it requires very high amount. And why is it low in case of ChIP-seq? If you remember, we had talked about template amplification strategies, all of which are able to increase the starting of material by different methods. So, yeah, what you see on the right, you have this is an example of the chip-on-chip protocol, which was performed for Drosophila melanogaster embryos, and yeah, so after formaldehyde fixation, chromatin preparation, IP, cross reversal amplification, Kleno labeling, you hybridize it onto a tiled array, and you perform statistical analysis to visualize and integrate your data. Just yeah. So next, when we talk about the sequencing protocol, what can you sequence or what should you sequence? You can either sequence, not either, you should preferably always sequence your input material, which is uh, the raw DNA, which is expected from the cell, the complete DNA lysate. Second is your pull down chip material. Third is your mock IgG material. And then when you proceed for sequencing, you see something like this, which is your chip fragment, which is flanked by adapters and then barcode. Just let me share my screen. It's getting stuck. Yeah. Okay. So when you proceed for sequencing, yeah, when you proceed for sequencing, this is your chip fragment, which you actually want the sequence for. These are your attached adapters, which we added during library preparation. And to that, you have your barcode. which allows you to identify where the sequence that you eventually get, the read that you get, where has it originated from. Now this is your read one and read two, because this is an example of a paired end sequencing, and along with it you have your barcode read, which is going to allow you to identify, sort of categorize your sample, and you end up with information like this, which shows you how much enrichment or how much binding there is at the site that you wish to study. And that is because here your protein was present to which your antibody bound and then pulled down those regions and then gave you all that information. Now, one very important factor to be noted whenever we talk about chip sequencing data is that it cannot give you absolute values. It always gives you a relative comparison. What do we mean by that? It means that if we simply got this material sequenced, only the chip pull down material sequence, all we would see is that okay, let's assume that we have designed very specific primers for our region of interest also. But even under those circumstances, all you would be able to possibly tell is that there is some binding which is happening, but that is going to be inconclusive in nature. Whenever you have to plot your ChIP-seq data or chip PCR data, there is a comparison that needs to be made. Either that comparison is with input, which basically means that um, in a particular cell, the total lysate, the total chromatin lysate that you get, within it, what is the percentage or what is the possibility that this DNA and this protein are interacting with each other, which basically mimics the original in vivo condition. And then in comparison, when you pull down your sample, then what is the expected comparison. And second way of plotting your data it's sort of a fold enrichment, which means that when you have not added an antibody, which is during your when you have not added your protein specific antibody, which is what happens in your IgG sample, then compared to the IgG sample, how many folds more enrichment of your protein of interest are you getting in the pull down sample, and that's how you plot fold enrichment. So these are the two common representations of chip data. So yeah, therefore, whenever you are representing ChIP-seq data, there has to be an external calibration. And relative to that, you can tell how much enrichment or how much signal is there at your protein of interest. So yeah, and then this is sort of what you end up with, that these regions have very high peaks. This region 1 and region 2 has very high peaks, indicating that this is where it commonly binds. Next, you have the different possibilities or the different types of enrichment which may occur. So commonly you would see for histone modifications like we just discussed, all the 5 core histone marks or some core histone marks can show this tendency whereby there are single points where you will get enrichment. Second possibility is when there is a protein which does not have a very sort of tight binding, but instead it usually does not bind to a sequence. It usually binds to a region, and therefore what you get as an output is a lot more broad, and usually that you find for pol 3 itself. Uh, and third is a type of enrichment which is completely sort of non-specific with each other, which can either be a false result or it might be a protein which binds everywhere in the genome, like for example histone H3. So yeah, these are the different types of enrichment and the types of plots which you expect. And if you see, obviously, you can see there's a very clear difference between the binding of a typical transcription factor and the binding of uh, histone marks, which are generating proteins. Okay, so till now, what have we discussed? The principle of chromatin immunoprecipitation, pulling down the sample, cross-linking, lysing cells, pulling down the sample, and you're washing it, obviously. Then you want to get your sample sequenced somewhere. After library preparation, and then finally the data that you get as the output of your sequencing project is what you're going to analyze, and that is what's going to give you conclusive, hopefully conclusive results. So if we consider that this is quality. So this is the first step, which is quality control. Again, if you remember, this step is very similar to what we had initially seen during our discussion on whole exome sequencing and RNA-seq data analysis. So first step is pre-processing the data, which is where you make sure that the data is fit for further analysis before investing too much energy in it. So this is quality control. Then that is done by fast QC tool. Then you have genome alignment, which allows you to map where exactly your sequencing read has come from. The output of this particular step will usually give you a SAM file, which you must convert into a .bam file. Then this needs to be sort of annotated. If you remember, we had discussed annotation the previous time, and that is usually performed by bead calling, peak calling, which is done to give rise to a bed file. The peak calling basically tells you where has that particular peak come from, and there are tools to do that as well. And then finally, you analyze the assessment quality of your chip experiment. You compare peak calls between groups, and within each group also you combine your peak calls and then come to a conclusion. Yeah. And what do you expect from this data? Basically, you want to A, look at visualize that data. B, look at the motif to which your protein X binds and whether or not that is maintained. And third is annotation and functional enrichment, that what is it eventually going to do? What is the impact, functional impact of that of the functional impact of this binding between this DNA sequence and this protein? Okay. Now, what do you need to consider? Let's say you have decided that you're going to perform ChIP-seq, you have prepared your samples, and you've sent it for sequencing. What all should you know? Before you even come to the decision that sequencing needs to be performed, the reliability of a chip analysis is governed by how good is the antibody that you are using. If you remember, I just said that you should A, always make sure that the antibody is suited for this experiment, and B, you need to make sure that there is a the quality of the product or of the isolate should also be very high. There should be as minimum false positive results as possible from any non-specific uh, finding also, and unexpected results should, if you do see that there is a result that you did not at all hypothesize, then in that case it needs to be validated using multiple antibodies. So what does this mean? It means that many a times in the under circumstances where you do not have an antibody that is monoclonal in nature for your protein of interest, sometimes people may also use polyclonal antibodies. Now, in that case, what is a possibility is that there might be some it should not happen, but it's a possibility that there might be multiple off target sites where the antibody may bind instead of your protein of interest, and in that case it might show you enrichment at those regions, which would of course not be the correct result. So therefore, to avoid such false positive data, also you should use multiple antibodies, depending on the availability of those, to validate your results. And if it is an expected result, then well and good, it aligns with your hypothesis. Even in that case, you can use multiple antibodies as well for validation. Now, while most ChIP-seq tools are designed for sharp peaks that are located at some specific genomic positions, like for example, at some transcription start site, some histone modifications, which are associated with large genomic domains that result in very broadly distributed enrichment regions. You have examples of H3K27ME3, H3K36ME3, which are already strong modifications that I've mentioned, which are found to sort of spread over multiple 100 kilobases, which indicates very large regions, while H3K9 ME3 peaks they usually expand to only a few MB or a few megabases. Uh, Among these, yeah, this is basically what it shows. If you see here, these are sort of point sources. Very sharp peaks are usually found. Mostly you will find it for transcription factors like CTCF, MYC, etc. And another example of this is also H3K4 ME3. Um, but they can also sometimes give you broad domains in some cases. You also have broad sources, like you can see here, you do not see uh, very polished peaks, but you can see enrichment over other sites, and that is seen for histone marks, like I just mentioned. And these enrichments, they indicate that these are, because they're histones in nature, they do not bind to one region and then completely stop, but instead they bind to a stretch or a range, um, depending on what is the indication of that binding. And sometimes you might see that enhancer markers like H3K27 acetylation, H3K4ME1 can produce sharp peaks, but sometimes they construct broadly enriched regions, which are called as super enhancers. So this, why are we talking so much about the nature of the peak? Is because this can affect the choice of the computational tool which you would use for your analysis. There are certain tools better suited for broad peak, some tools better suited for the other one. Specifically, I think for, if I'm not wrong, uh, at the end of the analysis, which we will discuss in more detail in the next class, we'll just begin the analysis protocol today. But I did not want to mix up so many things on the same day. But basically, at the end of your analysis, once you have annotated the number of reads which are aligning with uh, a particular region, finally a sort of statistical test is applied, which what whether or not it is significant over the input and the IgG sample that has been utilized. In that case, the type of analysis that the MAX2 most commonly used tool is called MAX2, MACS2. Um, when you are running that code, at that time you should mention whether or not the peak you expect is broad, or if it is a point or a narrow peak. So that is going to completely differentiate your results, because if you consider data like this, it will look like there has been no enrichment at any point at all, which is of course not the case and would give you false results. The third type is sort of a mixed sort of enrichment over input, and that is well, it's somewhere you would find a broad peak, somewhere you would find an enriched peak. So basically, you would see this for certain different types of factors like RNPOL2 or uh, activation or repress complexes, SUSY12 like complexes. Basically, this is possibly the start site of RNPOL2. So maximum binding would be possibly be present here, but an elongating RNA polymerase could possibly bind here, which is why you see a broad peak in these regions. So again, this biologically makes sense only because the enrichment that you see is over input, that compared to the input, this much enrichment is present when I'm specifically pulling down the protein of interest. So yeah, other examples are tools like ROSE, which is used to detect super enhancers. Music is another tool which can estimate the average sample peak width that needs to be investigated. Now, when we talk about ChIP-seq data analysis, uh, there are two possibilities that you can proceed with. Don't think I have a lot more slides after this. Let me check. Yeah, just two slides after this. I think I could have included more information, but it's okay. We'll wrap up the class a little early then. Yeah. So among the key steps which need to be followed in ChIP-seq data analysis, the initial part is quite simple. similar. So, of course, you prepare the DNA library, and then there are two types of sequencing methods that may be opted for. One is a single end method, the second is a paired end method. Single end method will sequence obviously the DNA fragment from one end, making it suitable for short fragments and making it more cost effective. The other possibility is paired end, obviously, that sequences from both ends of the same fragment separately, which makes it ideal when you are interested in analyzing very complex genomic regions where the nature of binding of a protein can also vary. In Gypsy, these reads most commonly are from paired end sequencing because yes, sorry, single end reads are more commonly used instead of paired end reads because DNA fragments typically are 200 to 600 base pairs, which are shorter than those used in transcriptome sequencing. But if you remember, transcriptome sequencing preferentially uses paired end data to make the data more, to have more confidence in the data. Now, although sequencing technology was limited to short read length up to 2015, recent advancements have now allowed for reads of up to 100 base pairs, and the number of reads depends on the target, and typically it ranges from 500 million to 5 billion reads, owing to the potential loss of DNA molecules under various conditions. ChIP-seq also sometimes requires pooling a very large number of cells, somewhere between 1 million to 10 billion cells, depending on the target. Sometimes you can have very abundant targets like RNF2, which is present in many regions of the genome, or histone modifications also can yield sufficient signals. But if it is a protein which is not as abundant, most commonly, say if it is a transcription factor that is not very abundant but is playing a key role, or it's just a protein you wish to study, then in that case, to enrich the signal, it's very crucial to have very large number of cells, and it's like how do I put it? It's another way of optimization that also needs to be done, because you want your sample to be as um, abundant with the protein of your interest as possible. However, when you lyse a cell, you cannot specifically, initially, you cannot specifically enrich only one type of protein. Instead, you'll have a large number of proteins also if you use a large number of cells. Now, in that case, you have to dilute this lysate in a manner such that when you add your antibody, the antibody is not sterically hindered or is not competed by the presence of these large number of proteins, giving you non-specific incorrect results. But instead, the peak that you get is correct. And therefore, you want there to be sufficient cells, but you do not want it to impede with the antibody's actual function, so that its epitopes are not concealed, or that it is not going to non-specifically bind and basically can do its job correctly. Now, yeah. However, for low abundance proteins or histone modifications, which are very largely distributed across the genome, larger number of cells are recommended so that you can get accurate quantification. Appropriate negative control, therefore, is very crucial during IP, and input controls are also very, very crucial because you need to perform read normalization to reduce the background noise. So the first step, even in ChIP-seq data analysis, is after you have performed, or after you have outsourced your samples, sequencing has been performed by some company, they have gotten back to you with the results. What they'll give you initially are raw read files, much similar to the transcriptome data file, and you have to perform QC. So QC, if you remember, quality check or quality control was first performed by a tool that is Fast QC, which is basically going to overall analyze your sample and give you certain comments on it. Following which this also includes sequence composition, or like whether or not there's a bias in the sequence, or what is the mix, or what is the percentage of each of these bases in the complete sequence. Then you have raw sequence analysis, including overrepresented sequences, which includes most commonly telomere, which is what you see here. And after that, you also use a tool which is going to trim your data. The example given here is of Galore. You can also use Trimomatic or any other tool which is able to do the same job. Again, each of these these tools have certain differences, but the overall idea remains the same, whereby you want to trim and get rid of adapters, get rid of any other labels, any other markers, and you want to specifically you want to remove the adaptive contamination to make sure that your read sequence is correct and yeah, it's correct. Let me just put it that way. And yeah, I think that is the material. Yeah, I had prepared for today's class. If you have any doubts, let me know, and then I'll just quickly show you how you can retrieve ChipSeek data, and then we'll end today's class in 10-15 minutes. So just let me know if you have anything you want to discuss.
No, ma'am. Till now it is clear. Like you can go ahead. I'll just watch the video once, and if I'll have any doubt, I'll ask.
Okay, sure. Just it. I think I had shown you guys how to retrieve RNA sequencing data, if I'm wrong. But basically, what you use is the Geo database, NCBI Geo. Oh, let me share my screen. Yeah. So you use the Geo data set. So this repository is immense. Please make sure you make the most of it. Um, Basically, the idea is that you can, if you have studied from a particular paper, whereby you want to study some protein of interest, and you have some information about it, and at the end of that paper you will find the GSE ID, which is a geo data set ID, basically to give you that information. So at the end of any paper you will see the data availability statement present, where you'll get RNA-seq, ChIP-seq, ATAC-seq, whatever sequencing they have performed, you'll get all of that information. But apart from that, let me just randomly search for any protein. Um, I'm searching for AR, which is one of the proteins we work with in the lab. AR is the androgen receptor. And I'm going to just put these two keywords. Yeah, you can put these in quotes to make the search more specific, etc. But yeah, you can go ahead either way. Uh, some other, basically at the end of these techniques, you will see whether or not it has what sort of data it basically has. Let's look at this first one. This, yeah. So when you open any particular data set, you will always find this information about what type of experiment has been done, what were the conditions that were maintained in that at that point of time during the experiment. And here, for example, the overall design suggests that ChIP-seq has been performed for this particular protein, AHR, and H3K9 acetylation in some control cells, which were some sort of some variant, and ChIP-seq of AHR in the knockdown cells. So in control versus in knockdown, they have performed ChIP-seq of this particular protein. So this was, you'll get all the information about the person who has submitted it. And then when you come here, you will see, yeah, you will see this sample. Now, if you look here, you'll see you have chip, you have input, you have chip, you have input, you have chip, you have your input. And the conditions also are different here. You have control shRNA, input and chip. You have control shRNA, input and chip for H3K9 acetylation. And you also have the same for AHR, which is the other target in the other cell line where there is knockdown of this particular gene. Now, if you see here, in most cases, people obviously do not get IgG sequence. It will be a waste of money. It's sort of a negative control. What you can do instead is that whatever information you have gotten from here, you can sort of download that. Not download that, you can validate that. I mean that let me put it in a different manner. Say, for example, between these two cell lines, once you have performed the chip sequencing between the control, between the knockdown and the control cell lines, whatever comparison you have made, you can validate that using qPCR. And if you have your IgG samples with you, you can also cross check the same in IgG to make sure that there was no contamination. But normally people do not get this sequenced. So yeah, so from here, if you open any random experiment, let's see, you will see all of this information, the type of antibody used as well, whether there was any conditional variants, what was the growth protocol. So anytime you wish to do an experiment similar to this, you can look at all of these measures and then make sure that whatever you wish to validate also follows similar protocols. Otherwise, the reviewer might ask why you have chosen protocol different from the same, and you have to justify it. So yeah, the extraction protocol is also given under most cases. Here you'll see cells are fixed with 1% formaldehyde, quenched with glycine. Yeah, this is another step. It is usually not something you'll find in flowchart based protocols, but when you have your actual lab protocol, you'll see this is something that is done because formaldehyde can also continue to keep on this cross-linking reaction to sort of stop that. You add glycine and quench its activity, and then wash with cold PBS, release nuclei by the buffer, perform resonication. Then chromatin is also digested in this case using MNAs, and then you release the nuclei by sonication. Purified chromatin can then be utilized. And here you can see this was shipped to some company called Novogene, and sequencing was performed. Your instrument model is provided to you, and then you come to the data pre-processing steps. So we'll go over it quickly. The first step, yeah, apart from that, you see the library name is provided. Then your data processing involves the use of Trimomatic, which is used to remove adapters and trim low quality bases. You get rid of leading, trailing ambiguous, low quality bases after adapter clipping. Again, if you remember when we discussed the properties of Trimomatic, how to adjust the parameters so as to not lose out on relevant information and yet remove contaminating data, that optimization needs to be done. Then comes Bowtie 2, which is used for read alignment. This is a different tool, which is something we'll come to in our next class. I think in RNA-seq we had discussed STAR and STAR Aligner, HiSat2. These are commonly used for transcriptome data alignment, but Bowtie 2 works on a different principle. It cannot recollect if we discussed about I2, but most commonly it is not used for RNA-seq data. Usually it's for ChIP-seq. It aligns to the human reference genome. Here, HG19 has been mentioned, but you should preferably go for HG38, considering it is the most recent release of the human reference genome data. And after alignment, unique reads are utilized in the subsequent steps and then used for following downstream analysis, which involves peak calling. So the max2 peak calling, if you remember, I had mentioned MACS max2 for peak calling of the aligned data. And these parameters basically tell you how significant the data is. Again, like I mentioned, the BAM coverage utility in the Deep Tools. Deep Tools is another software. It converts this BAM file. Again, if you remember, we had talked about SAM file being converted to BAM file, which is more user friendly. Apart from that, there was also a genome index file, which was BAI. BAI file basically sort of profiles or indexes the entire genome data so that it becomes easier for the software to scan through and interpret whatever information you are providing to it. So the R packet chip seeker is finally used for peak annotation. So this is the protocol they followed. There are ifs and buts in this. You can use it as well. And most commonly, you would find the completely analyzed file also. You can get the big files directly and you can directly visualize them in the software, which is known as IGB, Integrated Genome Viewer, or IGB, Integrated Genome Browser. That's something you can use. Again, we'll go into more details about this in next week's class, but this is something that can be done. And max2 peak calling is also provided along with it so that whichever region you're interested in studying, you can be sure that the information that you are getting um, like the information you are interpreting from the results are statistically significant. So all of this has been provided. So again, like you can see, this is very recent data on Feb 28. The last update has been provided just like 2, 3 days ago. So from here, either you can download the big files along with the peaks from here, or what you can also do is that you can copy the SRA ID and then you can download the ChIP-seq data file separately. So let me just check the, just give me a minute. I'll check and get back to you. Yeah, just right here. Uh, yeah, from ENA, I think I used to download it from. Yeah, so there are two possible ways in which you can download that. Either you can go here and get the SRA file, the SRX file, which is experimental detail. Uh, from here, you can download the same. And as you can see, these are quite large files, 3 GB just for one sample. And if you're comparing multiple replicates here, you saw there was only one replicate. Usually people preferably get two done so that you can be sure that your data is not skewed in any manner. You can get three also to make it more easier to calculate stats. Um, but yeah, these are quite, I'm not going to download it because it's immense and I have no use with that data, but you can do that. And the other thing you can do, that I think it was the SRR ID that I possibly need. Just give me a minute. I think this is where I need to get it from. So you can search your SRR. You can search or view it however you wish. Uh, able to detect it. Research the SRX then. Okay, I think this has not been uploaded on the ENA browser. That's why we're not able to detect it. But let me try a slightly older one, which possibly has been, let's check this. This is a little bit slightly older, but so basically, these are different repositories where this information needs to be uploaded. Yeah, so this one we have here. So this gives you all the information here as well. It's just easier to download from here. That's why I go for it. And you can see this is your FASTQ file, fastq.gz, that you can download from here. And this shows you, you always should cross-validate that the SRX and the SRR files that you have seen in your original repository are what you see here, and this you can download. I hope it has not started yet. No. So that's what you can do. Um, so yeah, apart from that, you can simply view the data, whatever information is available, you can go ahead and check that. And in this study, if you see, yeah, in this study, they have taken two replicates, each of the input and of the pull down, because that makes it a more reliable data set. And that is what you will also find for most data as well. So I think that is pretty much what I can show till now, because we have not proceeded forward with it. But here also you can see you would find the big files available. You can download it and you can directly visualize it on IGV. But if you wish to change any parameters, or if you just simply want to learn ChIP-seq analysis by yourself, which is something you should preferably know, considering if you are in academia, it's something you'll come across or probably in the long run. The two strategies that are commonly used are either using a common online tool called as Galaxy, or if you have have access to a Linux system, then you can run your own codes. It just requires installation of a number of different packages, a number of different softwares. But once you've standardized it, it's going to be very easy. And it's going to give you a lot more control over analyzing the data in a very free will form. You can optimize conditions based on what you expect, and you can do a more thorough search of your data. So, yeah, that is pretty much it. So, if you have any questions, you can ask, and then we will wrap up today's class and we'll meet again next Tuesday.
This portion was new to me, ma'am, so like I have to look onto it.
Okay, yeah, this I just showed you how you can download chipseed data. That's all. So that, eventually when you want to do analysis, you can do that. Either you can use their whenever people submit data, their pre-analyzed data can also be downloaded, which is what I showed using the BW files, the big files, or you can download their raw files, the first few files, which I showed towards the end, and then do the complete analysis yourself. So both of them are options that is like based on your comfort and based on your experience, you can choose that. But yeah, pretty much that is it. And once we'll discuss the entire analysis pipeline in the next line, you'll understand it in better detail. But that is pretty much it, what I wanted to cover for today. In the next class, which will be on 10th, next Tuesday, we'll wrap up gypsy analysis. And yeah, the next couple of weeks, I'll have to take extra classes because I think we have finished 17 classes, possibly 17 or 18 of them, I think 17. So we still have around 8 classes that I need to cover and that we have to do within the next month. So yeah, that's what we'll do. And there are a lot of topics that I need to cover as well. So we'll do that as soon as possible, and I'll keep updating you guys over email. And if possible, I'll try to take another test also because I think it is necessary. It helps, but I really hope that there's a better turnout this time. But let's see. Let me know if there are any issues with the scheduling, and we can discuss that. But yeah, that is pretty much it from my side. And if not anything else, then we'll wrap up for today. So okay, so I'll end the session for today. Thank you so much for joining. I'll upload the lectures by next week. I want to complete chipseek analysis and then I'll upload everything till now. Have a happy Holi and I'll end today's class. Thank you for joining. Good night.
Thank you, ma'am. Happy Holi, ma'am.
Okay, thank you. Bye.